Hello and welcome to another Piper Pod. As promised, we are going to move on to part 2 of the special senses, and this time we're going to be focusing purely on the eye itself. Now, the eye is a pretty amazing organ as far as the special senses go and sensory organs go. It's responsible for housing around 70% of all of our receptors. So all of the sensory receptors found in our body, the eyes contain about 70% of all of them. So as you could imagine, they are highly sensitive, and they're also the most used of all the special senses for those that have eyesight. On top of that, there's links with evolutionary studies and psychological studies that say that the shape of our eyes and the way our eyes are formed has a lot to do with our emotional responses and our means of communicating with one another. So what I mean by that is, if we look at the eye in front of you and you look at the cornea, you look at the sclera compared to the iris size, most other mammals don't have that same ratio. Okay, so the same ratio of white to colour. So what it allows humans to do is we can tell wherever anybody's looking in the drop of a hat, and we often use eye contact, direct eye contact, to invite or start a conversation or get someone's attention. We just use eye contact and we can easily tell what's happening. Okay, so that's just an interesting side note for anyone that wants to know. So when you look at the eye at the front, you're looking at about 20% of the actual eye is on display. Okay, and it's housed within the orbital socket with all the surrounding muscles connecting to it, and it's made up of our three main areas. You've got the sclera, the iris, and the pupil itself. Okay, on the outer sides we have the lateral and the medial commissure. Now, the only reason I put it in this picture is so then you'll know which side's the medial side and which one's the lateral side. So on the medial aspect of the eye is where our drainage channels and everything start. So if we look at the lacrimal apparatus, we'll see that the actual lacrimal sac, if you will, on the medial aspect of the eye is not what most people call your tear duct. Okay, so a lot of people say when you get sleep in your eye that you've got a blocked tear duct, and they always look at that particular area. But as you can see, the lacrimal gland is up laterally and superior to the eye, and it will actually drain secretions, tears, over the top of the eye, and they'll drain through across the eye and then go into the lacrimal sac and then down into the nasal cavity. So we have these lubricating glands present in order to protect the eye from drying out, to remove any small foreign particles that land on the eye, but also it contains lysosomes as well. So the lacrimal glands works as our external immune system, because the cornea and the sclera itself don't have any communication with the immune system whatsoever. So we need the lacrimals, we have its lysosomes, to come along and kill any invading bodies as well. So that's what the lacrimal apparatus does, and that's how the connection runs. Now, obviously, within all this structure, the layer over the top is called the conjunctiva. So when we're talking about conjunctivitis, we're looking at the inflammation of the actual layers of the eye itself. Now, dacryocystitis is where we get a blockage of the lacrimal sac itself, so therefore nothing can drain out. So you get a build-up of particles and minerals, basically, in the corner of your eye. And that's what people often associate with sleep build-up. Now, eyes are obviously capable of a lot of external movement, and we've talked about this in the past, but this is just to give you an idea of the 6 muscles that attach to the eye and allow for movements. Now, as mentioned, there's 3 cranial nerves that are involved in the movement of the eye. Now, cranial nerve 4 is responsible for the superior oblique, and cranial nerve 6 is responsible for the lateral rectus muscle itself. All the other muscles, or the other rectus muscles and the other oblique muscle, is controlled by cranial nerve 3, the ocular motor. Okay, makes it nice and easy to understand. But all of this is essentially the connection points for all of those muscles that aid us in our movement of the eye. Okay, when we look into the eye, as you can see, the eye is obviously much bigger. Like I said, all that's on display is the 20%. We can see all the other complex structures of the eye itself. Now we're going to go through the different layers of the eye and how they communicate within our senses, but I just want to mention that there's two chambers, if you will, within the eye. We have the anterior chamber, and then we have the obviously main chamber itself, okay, or the posterior segment. Okay, now each of these chambers are filled up with fluids. So we've got aqueous fluid and we've got vitreous fluid on the inside. Now aqueous fluid is this very light fluid that aids in keeping the shape of the cornea itself, and then also aiding in light refraction. Whereas the vitreous humour is responsible for helping light pass around the actual globe, but also maintaining the shape of the actual eye itself. Now, like most fluid areas in the eye, you'll see that it will drain in and out of the eye in order to maintain the nutrients that are required. Another thing to note is obviously where the optic nerve is itself. That area there is a blind spot for the eye. Now, whilst we very rarely notice it unless you do some very tricky little tests yourself, it is a place where no photoreceptors are actually found, right on the base of the disc itself. So as mentioned, with the draining of the fluid of the eye, what normally happens is the channels open just in the aqueous solution itself, and the pressure drains out. Okay, all fluid comes in order to increase the pressure. Okay, so just like other fluids in the body is responsible for the inflow and outflow in order to maintain a certain pressure for function. Now when that is blocked, obviously the pressure inside the eye increases. Okay, so it causes ocular pressure. And what that does is it actually causes, that increased ocular pressure causes reduction in neural signals being available. So it starts on the peripheral side of the house and actually will crush the neurons and not allow impulses to go through. And then you'll get that general pattern of blindness that occurs with glaucoma. Okay, and that's actually, this is what causes glaucoma, is the inability for fluids to drain out of the eye. Okay, and that's what we're looking for. So obviously the first to go is your peripheral vision, and then all of a sudden it will come in like curtains closing basically from the outside in, is classic signs of glaucoma itself. So I just wanted to let you know that this is actually what causes your glaucoma. Okay, from there we're going to look at the actual layers of the eye itself. So starting right on the outside is what's known as the fibrous tunic. So this is where your sclera and your cornea are found. Now your cornea is the white sheets of collagen, okay, lots and lots of collagen all put together, and their role is to help bend the light around to different receptors. Okay, we'll talk about how the light travels through to those receptors as we go along, but that's what it does. It doesn't have any blood supply at all. So the cornea itself doesn't have any blood supply, but it is amazing at regenerating itself. So it can actually repair itself. It can feel pain. It can feel a lot of pain for us to do something about it. So pain receptors will pass things to the lacrimal gland to produce more tears to help protect the cornea itself. But it doesn't have any vascularity, so it actually has its own inbuilt like phagocytosis that would occur within the cornea itself. So it's pretty cool out alone. It's obviously what keeps the whole eye its structure. From there we move into the vascular tunic. So this is your middle layer, okay? And there's three main positions, and then obviously the pupil, which is the absence of any other tissue basically. So within the vascular tunic we have our choroids, which is a blood supply layer. Okay, so what it's very, very dark in pigment, so it helps prevent light from escaping out of the eye. Okay, that's what the choroid part of the eye would do. Now your cilia body, that's contained to the muscles, and that's what feeds the muscles within the eye itself. So your pupillary muscles that cause your iris to contract and relax is fed by the ciliary body itself. Now the iris is the colour, that colour circle around your eye, okay, and it's very, very muscular in its structure, and it has what's known as radial muscles that form around it. Now it's the contraction and the dilation of the iris muscles that actually form the pupil itself. So when we look at our pupil, as we can see, it's just this space in between those radial muscles themselves. So the pupil size and the iris contraction is controlled by the parasympathetic and the sympathetic nervous system. Okay, so if we have parasympathetic stimulation, it will cause the muscles to contract, and therefore the diameter of the pupils will reduce. But if we have sympathetic stimulation, it causes the radial muscles from the outside to contract, and it pulls the actual iris apart, and you get a much larger one. So it's two different muscle fibers that are involved in contracting and increasing and decreasing the diameter of the pupil itself. So when we do our Pearl test, that's actually what we are testing. The first part anyway is the relationship between parasympathetic and sympathetic involvement. Now on top of that, it is also linked to our emotional response. So if we see something that we are threatened or aroused by, that obviously will elicit a sympathetic response, and that will in turn dilate the pupils. And if we are relaxing and focusing on something really closely, then that actually will activate the parasympathetic nervous system, and it will cause the pupil to shrink in order to focus. Now that's a process known as accommodation itself, which we'll talk about in just a little bit when we start looking at the lens. Now the third and final layer, which is the innermost layer of the eye, so the part that touches the fluid, is your sensory tunic, okay, also known as your retina. So your retina is absolutely filled with photoreceptors. Okay, now photoreceptors obviously respond to light, and there's two different classes. There's our rods and there's our cones. Now our rods outnumber our cones about 20 to 1, basically, is what they say. So for every one cone, there's around 20 rods that are available. Now rods are responsible for allowing us to see in low light conditions. They will have really, really sensitive photoreceptive proteins within them. Okay, they say about 1,000 times more sensitive than the cones, and they are involved in picking up even the smallest amount of light, and they allow us to see things in low light conditions. Now these guys are found right around the peripheries of the eye itself. So if you're looking at an eye and right from the pupil you look right back, that is where you'll find the cones, and everywhere else which is peripheral is where you will find the rods themselves. Now cones, in contrast, there's only 6 million of those compared to 120, and they are responsible for our colour vision, and they are concentrating around the actual foveae itself. Now the foveae of the eye is a increased surface area, diamond shaped focus point at the back of the eye, okay, and it's where most of the light will be directed when we are trying to focus on something in detail. Okay, so that's where the cones are mostly focused around. Now if we lose vision to our cones, so if our cones get destroyed, then that's what's known as blindness or partial blindness. Whereas if our rods get destroyed, then all of a sudden we just have night blindness. Okay, so we can't see at night, but we can still see perfectly fine in high light or light conditions. So there's a difference that we'll find. So the rods and the cones obviously have a relationship with one another, and it helps us see things by being stimulated by light and then passing that message on. So let's have a look at how that actually works. So when we look at the inner cellular structure of the retina, we will see that light passes through the inside, basically. So where the end, if you will, of the photoreceptors actually are. So this is actually where the axon of the optic nerve is right at the base of the retina where the light hits first. So from there, the light has to travel through that aspect and then it's bounce off, if you will, the choroid. Remember, the choroid is the one that doesn't allow the light to pass through. And then from there, get picked up and stimulate the rods and the cones. Now when you stimulate the rods and the cones, what actually happens is each of them have a series of layers within them. Now each of these layers will have a different photosensitive protein that will be activated and travel down all the layers until the message, the action potential, is generated and passed on to your bipolar cells. Okay, so these are bipolar neurons, so they could be passed down and passed back from there. In there, the synaptic message is passed on to the ganglion cells, which again is passed on to the actual axon of the optic nerve. From there, the entire message that is being stimulated within the axon of the optic nerve travels all together until you move to the main cluster of the optic nerve in order to be travelled back to the back of the brain that we'll look at from there. So that there is simply how photoreceptors work, and it's a series of neuron chains all the way down to the optic nerve stimulated by light passing and refracting back through it. Now, a closer look at the rods and the cones, we need to basically understand what light is and what is actually being detected. So we'll look at the cones first, okay, because they're the easy one and it's the main part of our vision. So the cones are able, depending on the cone, able to pick up 3 different wavelengths of light. Now, most of the cones in our body are what's known as red cones. They represent about 60%, and then we have 30% green cones and only 10% blue cones, okay? Because most of the things we see within the spectrum of colour sit within the red range, and then we can focus down if there's anything outside of those spectrums. Now, what colour blindness is the absence of one or more of these particular types of cones. They don't communicate with one another. Okay, so as light travels through at a certain spectrum, remember light travels in waves at a certain spectrum, which is picked up by certain cones, therefore that particular message is passed on to one another. Now, if you want to test this yourself, this goes back to the difference between sensation and perception, is you can get a yellow light and a blue light and shine it in 2 different eyes and then ask the person what colour they see, and they'll come back to you as green, okay, because there's a mixture of those 2 spectrums coming in, which you'll be individually stimulating those 2 colours, but then your brain will interpret that particular colour as green. Okay, so that's the way we see colour, and that's the way that we can look at light. Now, your cones are incredibly receptive. Okay, they're not as sensitive to light, but they're incredibly adaptive to light. Now, this is why you can be in a dark room and automatically walk into a light room without too much trouble. Okay, you can see the light, you can see all the colours, that sort of thing. Now, what causes the pain when you wake up in the morning is just the sensitivity of your iris itself not allowing too much light in initially. Okay, that's what causes the pain, but you can easily adapt to light situations compared to dark, because in contrast, if you are in a light room and then walk into a dark room, your rods take a long time to adapt to an environment. So anywhere between 5 and 10 minutes it takes for your rods to actually pick up all the information from the low light environment, adjust your brain in order to be able to stimulate it faster in order for you to be able to see in the dark. And then as soon as you pop into the light, you'll readapt to light situations and then have to try to go back to adapting to dark situations. So the action potential generated by the rods is a lot slower than the action potentials generated by the cones. Now, the reason for that is the rods have many more layers to go through in order to pass the message on compared to the cones. So it actually takes significantly longer for rods to relay the information down to generate the action potential, and that's why we have the phenomenon of adaptation to light or dark situations. Okay, so let's focus on what helps us see and focus on things, which is obviously the lens. Now, lens is a transparent biconcave structure, so it's convexed on both sides of the actual lens itself. It's attached to cilia bodies, okay, so they can actually contract and relax in order to change the shape, and it's responsible for aiding in the refraction of light. Okay, so the things that don't really change that much is your cornea shape. So they can elongate and lengthen slightly, but It's a very crude sort of means of focusing. So your cornea shape, obviously extending the distance from the front to the retina or shrinking the distance, that happens. That's just a means of coarse focusing. Like over time, if you're reading something, your eyes will shrink, basically allowing close vision a lot easier. And then when you try to adjust, your eyes get fatigued because of it. But accommodation is something else. So accommodation is where we really hone in on something. So if we're trying to look at something far away or trying to look at something close to us, our lens shape will actually alter as to with our pupil shape. So this is what the term pearler is actually looking for. So we have pearl, pupils equal and reactive to light, but then we also have accommodating. And what accommodating means is if you have someone look into the distance until they're focused on something at least 6+ meters away, and then you get them to focus on something 30 centimeters away, and you watch their pupils constrict. This is a much finer control, okay, and it's not just a survival reaction of sympathetic, parasympathetic. It involves constant relay to the central nervous system and feedback back to the actual muscles controlling the pupil itself. So if you ever want to do pearl up properly, that is the way to do it. If you don't, then just write pearl. No harm, no foul. Okay? Don't get me wrong, if you write pearl up, most people will just assume that it's the one test anyway, except for geeks like me. So it's just an interesting thing for you to know. So when we look at lens accommodation itself, it's all about how light sources travel in. So our eyes are generally set for seeing something at distance. Okay, so anything that's about 20 feet away, 6 meters, is optimal vision for most humans. The reason for that is lights will actually cover into the eye at parallels. Okay, so there's no need for adjustment, it just comes straight in. If we try to look at something a little bit further away than that, then what will actually have to happen is our lens will have to elongate. Okay, we'll have to elongate in order to allow the focal point to still hit the back of the retina. Now, in contrast, if you want to look at something closer, the lens gets shorter and fatter, therefore passing the refractory properties down to the back of the retina itself. Okay, and that's the way that you adjust for seeing something far away and then seeing something closer to you. If someone's suffering from a condition known as myopia, then the refractory sight falls short. Okay, so this is nearsightedness. Okay, so myopia, the refractory properties will actually fall short. Therefore, the light message that gets picked up by the actual photoreceptors is diminished. So you're picking up less light. It takes longer for it to get to, which generally means you have a less crisp or less focused vision when you're looking at something if you suffer from myopia, because the focal point is not there, therefore the clear message cannot be relayed. In contrast with hyperopia, you will have the focus point being behind the retina. So by the time it hits the retina, it hasn't actually fully converged down because the optimal angle of refraction would be behind the retina. Therefore, once again, the clear message isn't passed on. Now, astigmatism is something unique all in one. This is where the lens itself is unequal. Okay, it's got a whole bunch of different curvatures. You've got an unequal lens, you've got an unequal cornea, which means when light comes in, you have multiple refractory properties within both of them. So the light mostly refracts from the cornea, then it refracts again from the start of the lens, and again from the back of the lens. Okay, so you have 3 different types of refraction. So if you have a mixed shape within those structures, then you can have numbers of focal points. And all that usually gets relayed as is you start seeing something that your brain interprets as depth. Okay, so you start to see the same item in depth, which generally means that the item that you're looking at becomes distorted, double vision or overlapping, that sort of thing. Okay, so that's what astigmatism is when we talk about it. Now, obviously these things are corrected by different shaped glasses for the first 2, convex or concave glasses for the first 2 to accommodate for the lens. And with astigmatism, Yeah, you can have glasses, or you can have laser surgery as well to essentially find out the shape of your cornea and your lens itself. Okay, let's look at the pathway of travel, all the interpretation. So us humans and most mammals, we have binocular vision. So a lot of times we look at something with both eyes, and that's what allows us to have depth perception. So we pick up on depth cues, and it allows us to estimate or experience a difference in things' depth. So we can generally calculate how far something is away and what size it probably is if it was closer to you, because of our ability to have depth perception. As whatever we are looking at emits light, which is what everything does for lack of a better term, it emits light towards your eyes will pick it up and relay a message to both sides of the hemisphere. So both sides of the primary visual cortex will get the message. So it will come in from your eye, depending on what part of your eye receives the main amount of stimulus, will allow our brain to know the direction in which the item is sitting, or how large the item is, or how dense it is. From there, as it extends across the eye, and the messages increase or decrease around it, the brain is able to interpret something's shape from there. Okay, now this is all done by having all this sensory input coming into our eye. Remember, 70% of our senses are firing in, so 70% of our entire body's sensory processes can be stimulated at once, and all of them can pass a message on to the primary visual cortex in one hit. Okay, so from there, the primary visual cortex needs help. Okay, now this is done around the optic chiasm, where the message is relayed through there and then through the thalamus to the primary visual cortex, but then also it gets relayed to the primary visual association area, and that's really where the analysis gets done. That's where all the breakdown of all the sensory input gets analysed and then interpreted by your brain by relaying it to memory centres and different parts of the brain for interpretation, emotional response, or some form of effect that we want to do. So that's, as I said, it's incredibly complex and involves the entire brain, but essentially the pathway starts in the primary visual cortex at the back of the occipital, and then it will pass messages on through association fibres to different parts of the brain to elicit second, third, fourth, 100th responses from there. Okay, so that is the special senses of the eye, focusing purely on photoreceptors. I hope it was at least interesting and you got something out of it. But until next time, take care.